So it's a great pleasure today to introduce today's three speakers for an update on what's happening in Module 2 in CBMM. So I'm going to be very brief because I want to listen to all three of these talented scholars. So before I do so, I just want to give a big shout to Dr. Jerry Wang, who many of you know, who's been working in Module 2 and just successfully defended his thesis today. So now he's our youngest PhD in the lab in Module 2 and probably in CBMM right now. And so congratulations, Jerry, if you're in the audience. So very briefly, just to recap, Module 2, we are interested in how we can flexibly use visual information to answer essentially an infinite number of questions on an image. So we take an image like this one, and in a very short amount of time, we can essentially make a lot of inferences and answer questions about how many shoes there are, who are they, what are they doing, where is Obama's food, maybe even what happened before, and perhaps understand why this image is meant to be humorous. Borrowing a term that was coined by Shimon Ullman, we think that this is done by the ability to have computational routines, visual routines that can extract visual information, but importantly, they can be reused in a compositional manner and in a flexible manner. So that's one of the key questions we're interested in understanding in Module 2. And so those include potentially a large number of different types of routines. And we're trying to make progress towards defining what they are, trying to understand what the neuronal mechanisms are implementing these type of routines, and trying to instantiate those into biologically inspired computational models. So today we have three talented young people that are going to talk about work in progress in this domain. Will is going actually the order I think is not correct here. We'll start with Mengmi first, and then GA, and finally Will. Mengmi is going to tell us about one of the most basic routines that has to do with understanding the role of attention and eye movements to sequentially understand a visual scene. Then GA is going to talk about the transfer of visual information to episodic memory and how our brains detect boundaries to segment information to form episodic memories. And finally, we'll talk about stress testing computational models to understand whether humans and primates and non-human primates are susceptible to the types of adversarial images that have been so important in defining the limits of current deep convolutional neural networks. So without further ado, I'm going to stop sharing and invite Mengmi to start with her presentation.
So, hi everyone, my name is Mengmi. I'm a second-year postdoc from Kreiman lab, and these are my collaborators. So in particular, I want to mention Shashi, who is a very talented college student. He's been doing majority of the work I'm about to present to you very soon. So let me first start by briefly introducing you what is visual search and why we need it. So visual search is a very challenging problem in natural vision. For example, sometimes we have to find our car keys in a living room, or we had to look for our friend in a party crowd. And here is an example where I'm showing the subject is asked to search for a calculator, and he's constantly shifting his attention as indicated in the yellow dot, and eventually he managed to find the calculator in the red box. The study of visual search has a long history in neurophysiology and psychology. So in this work, our goal is to put together pieces of evidences in neurophysiology and come up with a computational model for visual search, such that we could quantitatively assess their behaviors and compare those with humans in psychology. This is to help us gain better understanding of the neural circuits involved in visual search in the brain. So there are two fundamental properties in visual search. This is the first one, which is called search symmetry. So let me explain to you what I mean by that. In the first experiment, what you are saying is the target and the distractors differ in terms of their lighting conditions. For example, the target is receiving the light from left to right, whereas the distractors are receiving the light from right to left, which is in the opposite direction. Here is the second set of experiments, and the lighting conditions becomes vertical. So in psychology, scientists have already found that we as humans were better at searching targets in vertical lighting conditions compared with the horizontal lighting conditions. This is intriguing to me because it leads to the question whether human visual system has feature preferences. And if yes, like where does this feature preference come from? Is this simply because our eyes get used to seeing more target objects with the vertical lighting conditions? And is this simply because, the sun is always up in the sky and therefore we tend to see more target objects in vertical lighting conditions? Yeah. So this is the second fundamental property in visual search, which is called feature conjunction search. In the first row, what I'm showing are the conditions where the distractor and the target differ in only one aspect. It could be either in color, orientation, or size. In the second row, the distractor and the target differ in more than one aspect. So this is the feature conjunction search condition. And scientists have already found that we as humans were worse in searching in the feature conjunction conditions compared with the simpler conditions, where the target and distractor only differ in one aspect. So with these two fundamental properties of visual searching mind, let me give you an overview of our proposed computational model for visual search that is called eccentricity net. So same as the psychophysics experiments, the eccentricity net is first presented with a target image. Next, the eccentricity net is presented with a search image. We use the same 2D conv nets that have been pre-trained on image nets for object recognition to extract the feature maps. Then we could use those feature maps on the search image to predict the bottom-up saliency map. Similarly, we can use the stored abstract representation of the target to modulate those feature maps of the search image across multiple layers, and this results in an top-down modulation map. Next, we can linearly combine these two maps together to generate the overall attention map. Then we can use the vertical mechanism to choose the location with the maximum attention value on this overall attention map as the next fixation location, and this process iterates until the eccentricity net finds the target. If you have been to my previous talks, you are probably familiar with and think this architecture is very similar as our previous work, and that's true. But I want to highlight three very important components that are new in this current model. So the first component we introduced in this new model is the bottom-up saliency model. So what we did is, given any units on the feature maps of the search image, we compute their self-information. So the higher the self-information, the more salient that location is. The second component that is different from our previous model is this top-down modulation. So instead of performing top-down modulation at the top of the 2D conv net, here we are applying top-down modulation across multiple layers. The third component, which is different and new, is in this pre-trained 2D conv net. I'm going to talk a little bit more about the third component. So I'm pretty sure most of us are very familiar with the standard 2D conv net architecture. But here, if we zoom into the third pooling layer, so let me explain to you what I mean by this eccentricity-dependent pooling layer. So given any unit G on this pooling layer 3, we could compute its Euclidean distance from the center fixation location. The further the distance, the larger receptive field size unit G has, as indicated by the different colors on the right. All right, so here we are just reproducing the plot, eccentricity versus receptive field size plots from the Similarity paper, where they have taken measurements of the receptive field sizes in macaque monkey brains. there are two observations if you are paying attention to this figure. So the first observation is that within the same visual area, as the eccentricity increases, the receptive field size increases. And if we compare across different visual areas, then the neuron in V4 tends to have larger receptive field size compared with the neurons in V1, V2. So now let's see how the eccentricity net performs in this. So what we did is quite similar to the one they did on monkeys. So we present this black image with a white square on top of it to this eccentricity net. And we ask eccentricity net to fixate in the center of the image. Then we slide this white square across the entire image such that we could record the neural responses or the activation values for each unit in the eccentricity net. And from there, we could compute the corresponding receptive field sizes. And this is what we get. I would say qualitatively, like these three curves match quite well with the one on monkeys. Just to give you a better understanding of the receptive field sizes across different layers for each unit in this each individual pooling layer, this is how it looks like. All right, so since in this project, we are interested in studying visual search tasks, and we want to test whether eccentricity net could transfer those knowledge learned in object recognition to visual search. Therefore, we loaded the pre-trained weights on ImageNet in object recognition, and we did not do any fine-tuning or retraining in the pre-trained ConvNet. This is the visualization of the example scan paths predicted by eccentricity net. So if you're looking at the subfigure A, eccentricity net starts the search in the center of the image, and then it takes the eccentricity net only one fixation to find the target in vertical lighting condition, whereas it takes the model 3 fixations to find the target in the horizontal lighting condition. Similarly, we could get example scan paths in the feature conjunction search experiment. All right, so back in the old days in psychology, eye tracking technology is not available. Therefore, scientists use this reaction time in milliseconds to measure how long it takes for the subject to localize to find the target. And since our model is generating a sequence of fixations, therefore, we did another separate experiment in order to map the number of fixations to reaction time. And after we are done with the mapping, here's what we get. So the dashed line indicates the performance for the model, and the solid line indicates the performance for humans. The blue curve denotes the vertical lighting condition, the red curve denotes the horizontal lighting condition. And there are two observations we can make by comparison. So the first one is the red curve is always above the blue curve. That means for both humans and model, they are very good at searching for targets in vertical lighting condition compared with the horizontal lighting condition. The second observation is that if we compute the slope of the blue curve versus the red curve, then the blue curve is relatively flat. So that means the number of items on the display has little effect in the reaction time. This is true for both humans and the model. And these are the qualitative results for both humans and model in feature conjunction search experiments. And we could also see similar conclusions. That is, the blue curve and the green curve are always below the red curve. That means humans and the model were better at searching simple targets compared with the feature conjunction search conditions. And the number of items seems to have little effect in reaction time in the simpler condition compared with the feature conjunction search condition. In psychology, scientists also introduced another measurement that is called the search slopes to evaluate the humans and the model's performance over all conditions from all experiments. So here, what you are saying is the x-axis is a human search slope, in reaction times per item. And the y-axis is for the model. And due to time constraints, I didn't introduce you total of 6 experiments we did. But here I'm just, so here you could have a brief overview and see like what are the actual 6 experiments we included in the search symmetry condition. So there are 6 experiments in total and 2 conditions per experiment. Those are the corresponding 12 markers on the plot. The dashed line indicates the identity line, and the dotted line join the 2 conditions from the same experiment together. So most of the markers are lying around the identity line. That means it has a higher linear correlation values, and it seems the humans and the model matches pretty well. Similar analysis could be applied in the feature conjunction experiments. All right, so far I have been talking about the 2 fundamental properties in visual search. And most of the time we have been evaluating eccentricity nets on very simplistic stimuli, for example, searching keys among L's or searching for simple shapes. The next task we want to test is whether eccentricity nets could generalize in naturalistic settings. So these are the 3 experiments that we did from our previous project. These 3 data sets include object arrays, natural images, and the finding Waddle experiments. So the first evaluation matrix we introduced here is the cumulative search performance. And it's telling you what is the probability of finding the target given a fixed number of fixations. Although eccentricity net is only able to assess very limited information from certain parts of the image because due to eccentricity, however, its performance doesn't drop. It performs almost as well as humans. Then the second evaluation metric we did here is the scan path similarity score. It's telling you how similar between 2 scan paths in terms of their temporal and the spatial dynamics. We could see eccentricity net did a better job compared with our previous model. It has a higher score. The last aspect that we evaluated eccentricity on is this Saccard size distribution. And it's just interesting that without imposing additional constraints, such as muscular constraints, eccentricity net just has a similar Saccard size distribution as humans. So to summarize, our proposed model approximates humans in many different, in varieties of aspects. This includes search symmetry, conjunction search, and even in naturalistic settings. So yeah, I'm happy to take any questions.
Hi, Mengmi. Just a quick question. I'm curious how the fixation size distribution compares to the IVSM model, since you showed it for your new model, but not the.
What do you mean by the scan path?
Sorry, not fixation. The Saccard size. So the previous slide.
Oh yeah, we do have that in the supplementary figures. So for IVSN, it's the distribution looks totally different.
I see. Thanks.
Yeah.
I'm Mengmi. Nice work. I have a question about. So you mentioned that so the network was pre-trained on ImageNet, and then you observe these differences, for example, for the orientation in terms of reaction time and number of Saccards. So I was wondering whether so you expected all of that comes just from the natural statistics of the images on ImageNet. So if you train the model now by randomizing the orientations of the images, you would lose that effect. No, and if you and whether you have tried that hypothesis, just retraining the model, but now on rotated images on ImageNet and see whether you still have that effect.
Sorry, Tiago. So would you mind clarifying what do you mean by effect here?
So for the first experiment where you showed that to detect the saliency in the vertically symmetric object was faster than the horizontal mirrored object, right?
Are you referring to the horizontal versus vertical
Yes.
lighting condition?
Yes.
Yeah, that's a good question. So just want to rephrase your question to make sure I understand. So you're asking if we retrain this model using those rotated image nets, images from image nets, then would this asymmetry effect be different? Yeah, that's so this question is fantastic because I personally think this asymmetry somehow inherits from this object recognition task. And we haven't tested this, but yeah, definitely. I think this is an interesting experiment that we should definitely try. Yeah.
Okay.
But and I would expect it to be the opposite. For example, if we rotated the image 180 degrees. Yeah.
Well, I mean, I guess if you just rotate the image 90 degrees, it's obvious because everything is rotated. What I was suggesting is if you just now for each image that you train, you just randomly rotate it so that you break whatever preferred orientations you have in the natural statistics of the images. So you would expect, if you train the model now under that situation, you would expect this effect to be gone, right? To be no asymmetry in these orientations.
Yep. Excellent question.
Hi, Mengmi. Great talk.
Hey, Pramod.
So my question is about search asymmetry per se, because my sort of understanding of search asymmetry is that you have the same two objects, but you just make one of them target in one situation and make the same thing as distractor in the other situation, and you would find differences in reaction time, right? So that is what search asymmetry is. For example, search for O among Qs versus Q among Os.
Well, yeah, I agree. Yes.
But would you find something similar if you sort of use your model to test that?
Let me just pull up the slide that is relevant to your question. Yeah, so here, these are the 6 experiments that we did in terms of search asymmetry. But I would like to clarify, for example, in this L among T's versus T among L's, we do see this search asymmetry effect. Yeah.
Okay, cool.
Yeah.
Thank you.
And this also happens in the orientation.
Okay, thank you very much, Mengmi. If there are no further questions, then maybe we can move on to GA.
Hi, my name is Jia, so you can call me JZ. I'm a postdoc from Dr. Gabriel Kreiman's lab. So today I'm going to share some of the recent findings about this interesting neurons we found in humans that detect cognitive boundaries to structure episodic memories. So as we all know, our lives unfolds over time with enriched, dynamic, and multisensory information into a continuous experience. However, our memory about this continuous experience tend to be, we tend to remember this continuous experience as a set of discrete events, which serve as the building blocks for our autobiographic memories. And one example will be, we don't necessarily remember a 2-hour movie frame by frame, but instead we remember it as a set of salient moments or salient events as we described here. Then the fundamental question becomes then, what defines an event, or what determines the onset and offset of that event? There are numerous like computational and theoretical works has been proposing that the transition from the continuous experience into discrete mnemonic episodes relies on the detection of cognitive boundaries. So before I jump into this neural evidence supporting this cognitive boundary detection concept, I want to stepping back a little bit to talk about another type of boundary, the spatial boundary, which has been well studied in the rodent electrophysiological studies. So here I'm showing the example, or for example, border cells, which reported in 2008, the science paper from the Moser's lab, which they found the neurons in the hippocampal networks increased their neuronal activities when the rodents approaching the spatial boundary in an open space. And besides encoding the spatial boundaries in a fixed, in an open space like the walls here, they also found the neurons in the hippocampal network which are capable of capturing the structure information from a more complex spatial environment. So here I'm showing 3 grid cells, which their neuronal firing patterns looks like a grid. And when the rodent's running around in this open area. But now if we put in some walls in the same open area to creating this hairpin maze and asking the rodents to running from one side to the others, what we found is that the neuronal firing patterns, the grid-like patterns, right now were segmented into repeated submaps, with the highest firing rates occurring most likely around the turning points of these walls. So both of this piece of information suggesting that the neurons in the hippocampal network was capable capturing the structural information from a spatial environment. And more interestingly, a recent study from Tanagawa's groups in which they were training the rodents running around in the same spatial environment repeatedly, as showing here for 4 labs, they found neurons in the hippocampal network increase their response like strongly to a specific lab, irrespective to the spatial locations. And on top of that, moreover, that they found this kind of lab-specific rate mappings phenomenon was highly transferable, because when they put in the same rat from the square maze to a different spatial maze, the circle maze here, they found these neurons are consistently showing this rate mappings to the specific labs, irrespective to the spatial changes. So this finding is suggesting that neurons in the hippocampal network may be also capable of capturing some sort of like event structures of non-spatial scenarios. So all these findings inspire us to do this work, trying to figure out whether there's specific neurons in the hippocampal network actually signaling the cognitive boundaries in humans as well. So to address these questions, we developed this task, which has 3 different phases. So during the encoding phase, we present a series of clips without any sound to the subjects. And the key point here is for those clips, we invented different type of boundaries. So as showing here, we have the no boundary, which is just a clips with is a continuous shot from a movie without any visual cuttings or visual editings. And for the saw boundaries, we're referring to those scene cuts, which the editors are changing the camera angles, but the clips are still from the same movie. And for the ha boundaries, we manually created them by taking clips from 2 different movies and put them together. So as you may notice that both the saw boundary and the ha boundary contains these visual discontinuities, but only the ha boundaries actually has a jump in the narrative story here. And one thing to mention that since we don't have any real boundaries in the no boundary clips, we manually defined the middle of the clip as a virtual boundary just for the analysis comparisons purpose. So after subject watched all the clips, which are 90 of them, 30 clips for each no boundary, soft boundary, and ha boundary conditions, we evaluate subjects' memories about those clips they watched in 2 different ways. So in the scene recognition task, we extracted frames from the clips they have seen before in the encoding phase and the ones from the clips they haven't seen, and asked them to decide whether that frame is old or new. And in the time discrimination task, we extracted 2 frames which across different type of boundaries and presented side by side to the subject and asked them to determine whether the left or the right frames occurs first in the original clips they have seen in encoding session. And besides the binary choice, we also invented some sort of measurements for their confidence levels just to make sure they have a good quality of their memories. So far we have collected 20 patients. We have 20 patients with drug-resistant epilepsy performed this task simultaneously with the electrode implanted in their brains to record brain signals. So first, let's look at the behavior data from all these subjects. For the scene recognition task, if we plotted out subjects' memory performance in terms of their recognition accuracy, their response time, and also their confidence levels. Actually, for all three boundary types, we don't find a significant difference across them. However, if we take a closer look, for example, for the accuracy rate, if we splitting the trials within each boundary types based on the relative distance between the targeted frames we extracted from the clips and their relative distance to the previous boundaries, what we found is that the frames right after the soft boundary and hard boundary tend to be remembered better than the ones that are further away. And this kind of distance effect we didn't observe in the no boundary conditions. Then how about the time discrimination? So if we plotted out the same measurements for the time discrimination task in terms like accuracy, response time, and also confidence level, what we already can see is that subjects tend to have worse time discrimination accuracy, and they take longer time to respond, and also they have less confidence about their choice when they are trying to recall the temporal order of two frames that cross a hard boundary in the original clip. So it seems like the boundary are influencing both the scene recognition and time discrimination memory. Then what's the neural signatures are actually tied it to, or potentially can explain this memory effects we observed in the behavioral level? What in terms of the neural data, what we are using is we are recording the single unit activity from the human subjects using the neural links systems and also this hybrid micro and macro electrodes. So the typical like procedure is the subjects were going to have a pre-implantation MRI scan first. And based on that, the neurosurgeons and neurologists are doing some surgical planning to determine where to insert the electrodes. And during the surgery, they will insert the macro electrodes first and using it as a guidance to insert the micro wires. And after that, they will do another MRI or post-implantation MRI or CTs to help us to localize where those electrodes are. And here I was just showing the signals we recorded. So on the top is the raw signals. You can see this local field potential data and embedded with some ticks, which are the single unit data we recorded from humans. So by doing some bypass filters and some spike sorting algorithms, we were able to capturing the single neuron activities from the raw data we recorded. And among all the 20 patients we recorded so far, we detected approximately 1,000 neurons from different brain regions. And for the analysis and the results I'm going to present it to you as following, we are focusing on these three specific regions, the hippocampus, amygdala, and perihippocampoid gyrus specifically because they're known to play critical roles in the episodic memories and also boundary detections. So as I mentioned before, during the encoding session, subjects were watching a series of clips embedded with different type of boundaries. So the first thing we look at into the neural data is trying to see whether there are any neuronal activities is able to differentiate this different type of boundaries. So here I'm showing you is a raster plot, a neural data plot we are generating for each individual neurons we detected. So among all the neurons we recorded so far, there are two types of neurons we found actually signaling the different type of boundaries we mentioned before. So one of them is, as showing the example here, which we call the boundary neurons. As showing here, each dot represents to the activation of that specific neurons, and we color coded for a different boundary conditions here. And what you see here is that this neurons increase its firing rate right after soft boundary and hard boundary conditions, but not for the no boundary conditions. And we also observe another type of neurons, as shown in the example here, which it only respond or only increase its firing rate for right after the hard boundaries, but not for the soft boundary and no boundary conditions, which we name it as the event neurons or event cells. And so far across like 580 neurons we recorded in those 3 regions, we were able to find 42 boundary cells. And they all have very consistent firing rate, firing patterns, as shown here. For each row represents to 1 boundary cells we detected. So both of them, all of them are constantly response to the soft boundary and hard boundary, but not for the no boundary conditions. And also across all these 580 neurons in the middle temporal lobe, we also found like 36 event cells. They also respond very consistently only to the hard boundary condition. And this plot also has some interesting information here as well. If we look at the both these 2 cell populations, we notice that they either respond to soft boundary and hard boundary only, or both soft boundary and hard boundaries. So the question will be, as I mentioned before, like both soft boundary and hard boundary, they contains these visual transitions or the visual discontinuities during the clips, but not for the no boundary conditions. So the question will be whether this absence of the neuronal firing in the no boundary condition is due to this lack of the visual transition information in the clip. So to address these questions, we were plotting this neuronal activity from these 2 cell populations aligned to the clip onset and also the clip offsets, which also contains the same amount of like a visual abrupt visual transitions, because they are changing either from a fixation cross to a whole movie or the whole movie to a fixation cross. So as showing here, these 2 populations of neurons, none of them are actually has this the same amount of like strong firing rate increase as they show to the soft boundary and hard boundary condition. And another interesting information in this plot is if we compare both the boundary cells and also the event cells, which they both respond to the hard boundaries conditions, what you probably can see here already is that there's slightly delay in terms of the response between these 2 cell populations. So if we further quantify this temporal relationship between these 2 cell populations by computing their average firing rate within each cell populations, what you can see here is that the purple, the pink line here is the boundary cells average firing rate traces, and also the purple one is from the event cells. So apparently these boundary cells are response a little bit earlier, with approximately 100 milliseconds earlier than the event cells. Then the question is, what the potential reasons actually driving the sequential firings between these 2 cell populations? To address this question, we delve into the spatial locations, or where those B cells or E cells are located in the brains. So as shown here in these tables, what you can see is that the majority of the boundary cells are actually located in the pure hippocampal gyres, while the most of the event cells are located in the hippocampus. And this kind of spatial specificity remains true when we extended our search to all the other brain regions which we have the access of the single neuron detections. So this suggesting this kind of sequential firing, where the early response from the B cells might be due to some the early response from the visual streams or visual areas, or the later response from the event cells, which are mostly from the hippocampus, might be the late comparable operations from the hippocampus. Then besides purely signaling what the boundaries, what different type of boundary it is, we also found this boundary cells and event cells are capable reflecting subject subsequent memory performance earlier on, even at the encoding session. So here, this is the same neuron, the boundary cells I showed you before, which increase the firing rate for both soft boundary and hard boundaries. If right now we're splitting this plot based on subjects' subsequent memory performance in the scene recognition, either they get correct or incorrect in the task, what we have found is that increased defined rate in the soft boundary and hard boundary condition are highly or mainly from the correct trials, but not from the incorrect trials. And if we further quantify this firing rate modulations across all the boundary cells, we see consistently this high firing rate in the correct soft and hard boundary trials, but not for the incorrect soft hard boundary trials. And also, we don't see any conditional difference between the correct and incorrect in the no boundary clips. And then how about the event cells? So this is the same event cells I showed you before, which increases its firing rate only for the hard boundaries. So we can convert this raster plot into a phase plot by converting the time when that spike activity happens relative to the theta oscillations we observed. So then right now this plot will be converted to a phase plot with the x-axis still be the time, but now the y-axis will be the theta phase. And if we further splitting this plot based on subject subsequent memory performance in the time discrimination, what we observed is that for the correct soft and hard boundaries, we observe this phase clusterings, but not for the other four conditions. And we can further quantify this phase clustering by computing the main resultant length, which as showing here, each arrow represents to the theta phase when that spike activity happens. So if all the spike activity tend to happen at a specific phase, which is the bottom plot showing, and we will expecting a very large mean resultant length as the blue arrow showing here, compared to the case like there's firing at the random phase, then we were expecting a shorter mean resultant length as showing in green here. Then if we further quantify this phase clustering effects across all 36 event cells, we see consistently this modulation of spike activity occurs in the correct trials for the soft and hard boundaries, but not for the incorrect, and also not for these no boundary conditions. So as I'm showing you this like B cells and E cells, they seems like modulated or using their spike rate modulations or spike time modulations to carry on specific memory informations from the scene recognition or time discriminations. And we also doing some different combinations to see whether they are actually representing to the other type of informations by different modalities. And we actually don't find any significant results. So which means that these two population of neurons, they are probably carrying this distinct memory aspects by their own individual strategies. So these are the single neuron data like responding to the boundary, different boundaries. Now we look into at the population levels, whether all the neurons in the medial temporal lobe also responds to the different boundaries. To address these questions, we formed this neurodynamics matrix by putting all the neurons, their spike data from all the neurons we recorded in the medial temporal lobe. And then we're doing some dimensionality reductions using the PCA analysis and downgraded this high dimensional information to a 3-dimensional space and plotted the neural trajectories at each time point when the subject's viewing the different clips. So as showing here, each dot in the lower dimensional PC space represents to the neural state at each time point. And when we are applying a sliding windows, we will be able to plotting the neural trajectories. And right now, which is plotting the neural trajectory from either no boundary, soft boundary, hard boundary before the boundary occurs. As you can see here, before the boundary occurs, they are kind of tangling with each other, not very closely. And if we extended our analysis windows by moving forward, which you can see that the soft boundary and hard boundary conditions after it occurs, which are marked by the black dot, the neural distance or neural shift shifted largely from the original states to a further away states. And if we further quantify this neural state shift by computing the neural distance, or what we call the multi-dimensional distance, as the Euclidean distance between any time point at the Euclidean distance between any time point to the boundaries, what we can do is converting this 3D plot into this 2D diagrams, and we consistently seeing this neural state shift in both soft boundary and hard boundary, which are the bumps here in both blue and red curves. And we further look at whether this neural state shift correlated with our subject's memory performance as well. So we plotted this, the accumulated multi-dimensional distance in the neural space and their correlations relative to the scene recognition accuracy and the time discrimination accuracy. Interestingly, we found a positive correlations between the neural state shift with the scene recognition accuracy and a negative correlation with the time discrimination accuracy, which potentially can be a neural explanations for the memory effects or the memory trade-off effects we observed at the behavioral level. So to summarize, at the behavioral level, we see the boundary enhance the recognition accuracy, and the hard boundaries actually impairs the order memories we have. And at single cell levels, we detected 78 neurons signaling different type of boundaries, either both the soft boundary and the hard boundary, or only the hard boundaries. And also the response strengths of those neurons are correlated with subject's memory performance in with different strategies. And at the population levels, we observed this neural state shift across boundaries, which positively correlated with the scene recognition, but negatively correlated with the time discrimination. So in the end, I want to thank all my my PIs and also my collaborators from our consortium teams and the funding source. And since the time limit problem, so I didn't present all the results we have, but they are public available in our bioRxiv paper right now. Thank you. So I'm happy to take any questions now.
I have a question. First of all, what a fascinating story. These are amazing neurons to have discovered and characterized. And I really like the controls that you have as well for the low-level stimuli. I had one question though. One thing is I'm trying to understand if I wanted to envision these neurons in the wild, right? In these patients' heads, when you're not showing the movies clips that are so well defined, what would correspond to the hard onsets and the soft onsets in the real world for these subjects? I could imagine that maybe you could have observed some of these behaviors, some of these neurons behaving when for example, you're setting up things and the subject's beginning to make memories related to maybe you coming into the room. What's the wild type equivalent of the stimuli?
Yeah, that's a very good question. Actually, we are also trying to delve into under more naturalistic setups, how these neurons will respond, or how we actually capturing those event transitions as well. So one idea we were thinking about, for example, when we are having these conversations, right? And suddenly my phone rings and I was trying to pick up the phone and starting talking about someone else. So I think it's something like this salient transitions or this jump in terms of the narratives can be potentially a hard boundary as well in a more naturalistic setup. But that's definitely harder to study in terms like a neural study or neural analysis part, because in order to have this digital powers to see those neuronal firings, that's why we created this manually like a modified hard boundary to just as a representative of those a salient change.
That makes sense. One thing that would also follow is maybe emotional content. Was there any kind of emotional content to the changes? Maybe something intense happening in one clip and then not in another clip that would also mark these boundaries?
Yeah, I think highly likely. So I think a lot of a behavior study has been done on that part of the scope, whether the salient information or the emotional related information can be a trigger for people to segment a continuous event. Behaviorally, there are definitely a lot of evidence to support this idea. But I think that's also a very interesting direction. We should also look at whether the neurons we found here actually represented that part of information as well. Thanks.
Gia, hi.
Hi. Hi.
It was fascinating, really, really wonderful.
Thanks.
I'm interested in the possibility that there may still be a low-level explanation, and maybe more generally, what is it really that that is driving the hard boundary neurons?
Yeah.
I mean, it's quite amazing. So I was, I can imagine a bunch of other experiments, but of course that means more experiments. So that's hard. But I'm wondering about things like color, not color, intervening times. Like there are movies with long, extended single take. The most famous is one called Russian Ark. It's an hour and a half, one take, and you could, move the time clips farther and farther apart, or is it, something like color, black and white, or something about the statistics of the scene, those particular clips.
Yeah. That's definitely a very not interesting. So I think we're all questioning about this topic, like whether this event segmentation is really driven by some lower visual features, or we were able to do that also without that. So I think some of the control we did here, I didn't show, but basically we were looking at, for example, the soft boundary and hard boundaries. We are measuring like a different, in terms of their visual features, whether the luminance, contrast, or complexity, or the color distributions, whether those lower features can be different between these two types of the boundaries that creates this different response in the event cells, which only responds to hard boundary, but not soft boundary. And we don't find any like a significant result for now, but it doesn't mean like nothing, all of this not contributing to the event segmentations. But I think a more interesting question you're asking right now here is how we gradually forming this event structure, right? Even without this very obvious visual cues. I think, so to referring to the papers like Anna Shapira's paper in Nature Neuroscience, she find like this event structure can also gradually form even though there's without any like a very transient visual cues. I found one explanations will be so the original like a predictional arrow series, like when assuming our brain is constantly doing some predictions, and if later on if that prediction arrows drops to increase to a certain levels, maybe that triggers this event segmentations already. So that can be fitted to this continuous scenarios as well. Yeah, but another thought which we haven't tested yet, but it's also in my mind, is that people has proposing this working memory and long-term memory relationships. So there's recent findings from Anna Jaffapur's studies, which she find that people tend to segment it in a continuous experience very frequently. They also have worse working memory performance. So I think that's a study she was trying to claim that your working memory capacity might be also indicators or factors like determining how you're gonna find segmented or like a core segmented same continuous experience. Yeah.
Great, thank you so much.
Thanks.
Can I ask one question?
Sure.
What do you make of the data preference spiking of those neurons? I forget if the prediction there is that they accumulate around a data phase in the recognition task or in the time order task.
Oh, you mean the event cells, whether this are the scene recognition or whether the encoding or the memory retrieval, right?
Exactly. So
Yeah, so this the dots, the neural activity I was showing here actually it's from the encoding. But I was just splitting their activity based on their behaviors from the retrieval part. So it seems like they were able to like reflecting or some sort of like associated with the their later memory performance during the encoding already.
Right. So, but what is the memory task that they're asked to do in this case?
Oh, okay. So in this task, basically we were pulling out two frames that cross either no boundary, soft boundary, or hard boundary, presented side by side to the subject, ask them to decide whether the left or the right frames happens earlier in the original clip they have seen.
But presumably there, the time is not, the time varies, right? So you could be pulling an image that happens 2 seconds after the transition, or only 1 second after the transition. Like it's not that you're testing, you're not grabbing an image that happens right around the time when you have that nice fixation around that data phase, right?
Yes, you mean like not right, the frame I take out and not necessary to be the frame right after the soft boundary or hard boundary, right?
Exactly right. So so the question, if that's the case, how do you, what do you think about this influence of data is in that encoding that is in a way unpredictable at the time of the test, right?
Yeah, so I think that's a good point. So another thing I haven't presented here, which might be related to the question you have here is, so for the retrieval data, we're also looking at the reinstatement of the neural context. So basically trying to see when people recalling this dynamic clips, whether recalling after frames or they recalling specific frames of this continuous experience. And what we are doing is basically computing these correlations between their encoding activity and retrieval activity, which we found is that we actually see a stronger correlations around the around the boundaries, but not around the exactly when that target frame occurs. So which we think, even though we presented maybe 8 seconds of the clip to the subjects, but what they really remembers are actually those frames right after the clips. And to be honest, for the clip itself, even though the clips are, the frames are further away from the clips, they are very similar to the one to the frames that after the boundary as well. So yeah, I don't know whether that addressed your questions or not.
No, so I'm thinking, is it possible that the one of the reasons you might be able to assign this data phases is that also data per se is a stronger signal in a particular case. Let's just say in cases in which subjects are really paying attention to the transition versus cases in which they're not paying attention, and there might be a correlation, and that you might have a low amplitude data, and then there your estimate of the phase is more inaccurate. Or that's one possibility. The other is that, is it possible that these neurons, in the cases in which subjects are paying attention, exhibit more of a time lock, a particular or a phase lock rather. That's, you're just capturing one instance of that in your measurement. So just different possibilities. Just if it were just pure recognition at the time of the phase locking, I'd have an easier time seeing how that happens. The fact that you have a time variability in the probes that you choose, that it's harder to interpret what this means.
Yeah, so I think to the first point you brought up, like attention, I think definitely attention is playing a critical role here in terms like subject's memory performance, and also this like event segmentations or the detection of the boundaries. But unfortunately, we don't have a very like sensitive way to measure the patient's attentions for this data set, but it's definitely interesting to see whether they're actually correlated with neural response or not. And the second point you brought up is for that theta phase, right? Or the theta powers, whether the stronger powers give us a better estimation for the theta phase, then we tend to have this like better phase clusterings.
Yeah.
That's something actually I'm looking into right now. So I think I should be able to give more updates later when I'm looking into this data. Yeah.
Yeah, cool. Thank you.
So thank you very much, everybody. Hector, also for the feedback. Can I suggest maybe I'm going to take an editorial decision now to move on to Will, because we still have yet another talk. But maybe, Jay, you can contact Hector, who has thought about this very deeply and a lot, and can get further feedback from him. Thank you. Thank you very much.
Thanks.
So let's move on to the last presentation today by Will.
Hi, I'm Will. So today I want to tell you about an ongoing project where we're trying to create adversarial images for primate vision. Is the slide working, and can you hear me?
Yes.
Okay, great. Yes. So this is a collaboration project with Lee, a visiting student from the National University of Singapore. And first, I want to explain why we're interested in adversarial images for primate vision and how we operationally define them. I'm sure most of the audience here know of adversarial images, but just to provide a brief background, adversarial images are really tied to convolutional neural networks, or CNNs, and they are some of the current best computer vision algorithms. However, if we add small, carefully crafted noise to natural images, CNNs can be misled to make wrong predictions. And here is a familiar example. On the left is an image of a panda, which is correctly categorized by a CNN. And in the middle is the added noise, magnified to be visible. On the right is the adversarial image, which the previously correct CNN now categorizes as a given. But you will be hard-pressed to see any change in the image at all. And what's more, this is not shown here, but CNNs assign very high confidence to these incorrect predictions about adversarial images. And this seems like an intuitively compelling example that CNNs are unlike our vision in being susceptible to adversarial attack. However, this conclusion doesn't really sit well with the finding that CNNs are very good models, the current best models for primate vision, shown by work including the one Mengmi just presented and from many other labs at CBMM. And I think there has been several roundtable discussions here about to what extent CNNs are actually good models of primate vision. And I think some here would agree that CNNs are good, but already quote unquote falsified models. But I still think there is no specific and satisfactory explanation of why CNNs can predict primate visual neural responses pretty well, but behave so differently when faced with adversarial images. So to talk about adversarial images for primates, including humans, I want to use the following definition. So I define an adversarial image relative to a clean image, which is a natural image that can be unambiguously classified. Then the adversarial image must be minimally different from the clean image, but results in a different classification. I want to unpack this a little more because this is pretty counterintuitive and contradicts some popular definitions of adversarial images. For example, a common definition is that the adversarial noise should be unnoticeable to humans. However, because we want to test adversarial images for primate vision, including humans, by definition, the adversarial noise has to be perceptible. And for the same reason, we cannot use the definition that adversarial images are images that humans do not get incorrectly, but for CNNs. Okay, another potentially counterintuitive consequence of this definition is that we can have trivial examples such as this. For example, let's take a clean image from a different class, x1, and construct this image, x0 plus the difference between x1 and x0. This, of course, is just x1 and will be classified differently than x0 by construction. And unfortunately, this will count as an adversarial image by our definition, although it's obviously a not very interesting one. So the key here is that it's really essential to be quantitative to control the size of the adversarial change. And to define what is small change, we really need to measure and compare to the actual separation between two clean image classes. So in this framework, adversarial image, to to a given limit, there will be so-called adversarial images for any visual system. But the key is how robust is, for example, primate vision to adversarial images. And can a small change of the image lead to a change in categorization? And here small is defined relative to trivial methods, such as just replacing the image. Meanwhile, however much perturbation it turns out we need, I think it will be interesting to compare that amount to what's needed to attack convolutional neural nets, both naive and adversarially robust ones, because I think that will be a useful benchmark for both computer vision and for models of the brain. And briefly, I want to summarize what's already known related to this question. There's a paper from Al-Said et al. Who showed that time-limited humans can be affected by the same adversarial images that affect convolutional nets. Their effect does require very short presentation times and basically reduces an already low accuracy of 75% by a further 10%. And this is not surprising because usually we are hardly able to see adversarial noise under normal viewing conditions. So for this to work, something has to be different. And another somewhat related result from Zhou and Firestone is that humans can guess what wrong categorizations will be made by CNNs. That doesn't necessarily mean humans will make the same mistakes, but just that they can expect how the CNNs will get it wrong. So what is not known is whether primates can be affected by adversarial images under normal viewing conditions, or more precisely, as we just discussed, how much image change is required to change the categorization. And finally, it is not known how visual neurons respond to adversarial images, both those tailored to CNNs and those tailored to the neurons themselves. And it's interesting because one prediction is that encoding models based on CNNs should diverge from actual neural responses when it comes to adversarial images, at least if primate vision turns out to be more robust than CNNs. So to try to address this question, here's our specific attack setting. We chose to conduct a targeted attack between human and monkey faces. And this is because we have access to face selective neurons, which are highly selective between these two categories, with clean categorization performance about 95% from just a handful of neurons. And I will today focus on the first attack direction. We also tested the reverse direction of monkey to human and non-face-to-face attack. And I'm happy to show those if time permits at the end. So to make adversarial images tailored to primate neurons, we designed what we call a gray box adversarial method. To many of you who are familiar with work from Puya and co. from the DiCarlo lab on neuropopulation control, this pipeline is very similar to their pipeline to make images that's designed to drive neurons. And the main difference is the loss function, which I will describe now. So to briefly just summarize the method, first we record the responses of some face-selective neurons to face and object images. And then we fit an encoding model of the neuronal response based on features extracted by, in this case, a ResNet. As I alluded to earlier, this class of models is state-of-the-art at predicting neural responses from arbitrary images. And in our settings, the fitted model explained about 50% of the explainable variance. And finally, we use this model, which is end-to-end differentiable, as a substitute for the actual neurons we want to attack, and use the model to modify clean images. The loss function then is to match the predicted response to the real response vector to the target class. So to be concrete, in human to monkey attack, we modify a human image so that the model predicts that the neurons will respond to the modified image similarly as to monkey images. So we call this method gray box only because we use some, but very limited information from the system being attacked. And to keep with the boxes analogy, in the literature on adversarial attack, this is probably much closer to black box attack, which assumes little to no information from the system, than it is to white box attack, which requires being able to differentiate through the whole model. And another reason this is limited information is because all the results I'm showing today are based on the same set of attack images made using the same encoding model, which was fit on data from one monkey. But we tested several monkeys and also humans. Okay, so now that we have this algorithm to make images, we also want to define the budget allowed for image change. And for this, we just used a simple metric, which is mean squared error of pixel value change. This is directly proportional to the square of the L2 metric commonly used in the attack literature. So the levels we used, we capped the MSE at 800 with pixel values ranging from 0 to 255. And we tested 10 linearly spaced levels from 200 to 800. And here are some example human to monkey attack images at these various noise levels. For reference, here's the distribution of the MSE distance between 250 each of clean human and monkey faces. The average of this distribution is about 6,500, and the minimum in all these pairs is about 950. And it might seem not too far from 800, which is the max level that we are using. But remember that adversarial attack has to work on the basis of individual images. So to estimate that, we can plot the minimum MSE as a function of number of images each image is compared to. So the little gray lines in here are individual images, and the purple line is the image average. It fits a power law, fit pretty well. And by extrapolation, it seems that we expect to need to compare one image to about 100 million before we come up with an image that's an MSE of 800 away from it. Okay, so this is, I just described how we attempt to make adversarial images tailored to neurons. We also, as comparisons, tested several other attack methods using the same noise levels. The first one we tested is adversarial images made to fool a pure model. These were made for the same fine-tuned ResNet that we used as the backbone for the substitute model, but this is without fitting to any neural data. Next, we also tested merged images, which are just simple linear interpolations between two clean images from the two classes. And thirdly, to control for image quality degradation, we also tested adding Gaussian noise. This isn't really an attack method because it's not targeted, but I think they serve as a good example of like how big the noise level 800 really is. And lastly, I'm not going to discuss the PS noise images, just putting it here because it's going to show up on the plots. And finally, I want to describe some results we have. First, we tested the attack images at the neuronal response level. For this, we recorded from face-selected monkey IT neurons to the images while the monkey is fixating. And to quantify attack success rate, we trained linear SVMs to classify neural responses to clean human and monkey faces. Accuracy on clean images were pretty high, as described earlier. And we're going to use these SVMs to classify the various attack images. So first, I'm going to show one example array recorded from one monkey. This is a UMAP visualization of the responses of maybe 20-something neurons. And you can see that the clean human face images are clearly separated, whereas the human to monkey adversarial images are already less similar to the human images and more similar to the monkey images. And of course, one caveat of UMAP is that it's not linear. So when quantifying the attack success rate, we just use a linear SVM directly on the normalized firing rates. Here, success rate is defined as the fraction of images out of 40 per condition per noise level. And the dashed gray line here indicates error rate on clean images. So you can see that the error rate, the neuron population is highly selective between the two categories. Meanwhile, the gray box attack images were able to basically successfully change the image categorization up to around 40% of success rate. And all the other attack methods didn't reach success rates much higher than the error rate on clean images. We tested the same images on 2 other monkeys, and these are the results. Gray box images in all cases were consistently more effective than the other attack methods. And interestingly, they achieved about the same success rate at the highest noise level, despite being tested on different monkeys using a model based on responses just from the first monkey. And just quickly, we verified one concern that the attack images could be misclassified by the SVMs simply because they became non-face-like. And this is not the case. As you can see from this UMAP visualization, we also trained SVMs to classify between face and non-face images, and almost all of the attack images are classified still as faces. Okay, so next I want to show some behavior results. And for this, we tested monkeys who were trained to do this binary classification task in cage on a touchscreen. And we also tested the images on human subjects on Amazon Mechanical Turk. So here are some results from the monkey behavior experiments. Again, the success rate is the fraction of images that were categorized as the opposite class. And the inset here shows each dot is an individual image, and the inset shows the fraction of trials on which the image is chosen as each class. Again, the dashed gray line here shows accuracy on or error rate on clean images. And the monkeys were well trained on this task. And still they made up to 40 to 60% errors on these gray box images, while not making as much error on the other attack methods. And finally, we also tested the images on Amazon Mechanical Turk. Oh, one thing important that I forgot to mention is that both monkeys and humans had a full second to examine the images. So the viewing time is practically unlimited. And it's interesting that gray box images were more effective than the other methods, even on humans, although the overall success rate is lower than the success on monkeys. Okay, to summarize what I talked about, I want to recall the question at the beginning, which is how robust is primate vision to adversarial attack? And if you only take one message away from this talk, I hope it is that it is not insane to ask this question. This can be a well-defined question and can be useful. And I would like to argue that this question needs a quantitative answer. Okay, so as for our results, based on our attack methods, we found that relatively small changes to an image could change the categorical representation of visual neurons in IT, and could also change categorization by primate behavior, both in a targeted way. And here, relatively small is compared to the natural separation between two categories of images. The actual changes are still very noticeable, but that's by definition required to change primate perception. And lastly, how does the required noise compare to the noise needed to attack CNNs? And for this, I would like to hear your thoughts and maybe get some pointers to literature, because I'm not really familiar with the state of the art here. And I just put here 2 arbitrary recent papers on the topic. And as you can see, the relative range of noise level is quite different, showing that the number can strongly depend on the task, probably on the image size as well as on the number of alternative categories. Regardless, I think being able to measure robustness in the brain and finding a fair comparison between the brain and the CNN is a useful yardstick by which to evaluate progress in making adversarially robust models. And finally, I just want to say that the success rates and noise levels we we could achieve is dependent on the methods we have, which is still fairly simple. So it's possible that future methods can come up with even more effective and even less perturbed images that change human perception. With that, I would like to thank my collaborator, Li, as well as my advisors, Gabriel and Marge. Thank you for listening, and I would love to hear questions and comments.
Well, quick question. First of all, excellent line of investigation. It's very, very cool. Very gutsy. I like it. I had a quick question. Do you have any, I'm just curious what the basic level PSTH responses of the neurons were to faces, human faces, monkey faces, and to the adversarial images. Were they very different or?
So yes, several answers to your question. First is what I already have, and just limited by what arrays we currently have in the lab. One of the recording has relatively lower SNR, as you can already see here, but overall, these are phase selective arrays. If you average images from the category, they are highly phase selective. And if you look at individual images, then the lower SNR array is less good. But just as I was making this talk, I was reminded that it will be interesting to actually test what I promised, which is fit models on clean images and evaluate, how well they predict adversarial images, as well as, just, as well as how the neurons respond to the adversarial images themselves, which I actually have an answer for. So this is the slide that maybe I passed over too quickly, but the adversarial images, they are little changed from face images, and they are still perceived as, categorized as face images based on neural responses. So presumably they are highly activated.
But in terms of overall firing rate, have you visualized just the PSTH?
Oh, I haven't. No, I have not actually. Yeah.
Okay. Interesting.
I really should, to look at the time course, for example.
Hi, Will. Did you say something about human responses or mechanical Turk? Was there some?
Yes. Maybe I went over too quickly over this as well. But the dashed lines here are just replotting the lines from the left, which were tested on monkeys. So we also did this on Mechanical Turk with two alternative forced choice task. And I guess 30% of the images were classified over subjects as monkey, the human to monkey attack images.
But it's, okay, so that's a forced choice paradigm. But, actually, I think if we're looking at it, we would say, well, that looks like a doctored monkey face, or that looks like an altered human face. It looks weird. It's neither, or I'm trying to figure out what it is that you will think is closer, or something like that. I mean, that would be my immediate response to these images. So I'm not sure. I'm not sure quite how to think about two alternative forced choice task in this case.
Yeah, I think that's an excellent question, though. In defense of the quantifications, I guess that's because, like in adversarial attack literature, it's always an n-way forced choice. But of course, it's an interesting question what these images exactly are doing. And I've been discussing this with Gabriel, for example, is the attack certainly is destroying certain like face features in the human faces, probably making them less human-like. But it's unclear to me, are they just making them less human-like, or is there some direction in which like it's also moving along to the monkey direction? The neural response data that I was just showing shows that it's not moving maybe too far away from the face cluster at least. But like of the vast space of possible pixel combinations, how tight are the islands of face and monkey images? And is this really in the no man's land? And maybe just to anticipate a little, I think a related question I've been puzzling over is that I feel the method, if we give it unlimited noise, it's still not going to come up with a full monkey image, right? So that doesn't mean that models are not good local approximations of the decision boundary, but yeah, that's just something to keep in mind as well.
Thank you. It's great and fascinating. Yeah.
Oh, just to quickly follow up on that, Will.
Yeah.
So yeah, it's pretty cool. So kind of in this slide, it kind of shows like you're not doing a fast gradient sign method attack or PGD. Like you can't do that as far as I understand, because you're working with a monkey, not with a network, right? I could be wrong, but I think that's what I understood. So here what you're showing is kind of the different strengths of one specific direction in which you're perturbing the stimuli, right?
Yes.
Okay. Yeah, it'd be interesting kind of in the PGD way of thinking if there's somehow like a curve instead of a straight line that you could draw in this high dimensional space. And if it is a straight line, maybe you won't get like, a monkey face, but if it is a curve, or if it's something that's not straight, you could maybe actually see something that is more natural. I don't know. It's just something. When I saw this, it resembled a lot the interplay between fast gradient sign method and PGD, that it just got me thinking. And I don't know what your thoughts were on that maybe as well.
Yeah, I just want to quickly clarify that this is based on actually fast gradient sign method. So okay, to do that, we had to basically make a substitute in silico version of the monkey neurons. And like an exciting direction is to be able to do this like zeroth order without an intermediate substitute model. I'm still not clear like how easy that is, because to be able to change images pixel-wise, that will entail a very large search space, right? Mm-hmm. So yeah, I'm just not courageous enough to undertake that yet. But unless we have a perfect model, I think to find the optimum direction of change, it'll have to be directly based on the neural responses.
Right. Yeah, kind of reminds me of like the eigen distortions paper by Berardino and Simoncelli. Yeah.
Exactly. So yeah, I also think of this in that framework, except that eigen distortion, I think, is only about perceptible differences, right, and this is both perceptible and targeted, so a little different.
Cool. Thanks, Will. Thank you, sir.
Yeah, no problem.
Hi, Will. I really like your talk. So one thing I feel is instead of framing your study in terms of adversarial attack, can you frame it in terms of like the minimal sets of features to recognize certain objects? Like, I think your human face turning into like a monkey face and leads human and monkeys to judge it otherwise. It represents like the image definitely has some features that our human or monkey use to do the classification. So this result, can it be interpreted as your adversarial attack definitely capture those minimal features to do the classification? And the follow-up is, if you do not start from like human face, but start from like gray screen or gray noise and just evolve or generates those features, I mean, can you still achieve those kind of performance?
So the first point is very interesting, and I have to think more about it. I guess the results as they currently are and the perplexing degree of this question basically makes there to be all sorts of ways to frame this. Like another suggestion I've heard is that this is finding a direction that it's like using a model to make microstimulation, for example, that could change behavior. So I'm not clear what is the best way to frame this, but your suggestion is the first time I've heard, so I'll have to consider that carefully. And the second one, I have some data related to it, which is the non-face-to-face attack I alluded to earlier. I hope that's relevant to your question. Sorry, I have to find the slide. So we did try to, it didn't start from a gray screen, but actually we're doing the experiment right now starting from not objects, but like noise images. And so use the same pipeline to make the images have face neural responses. And we could make that as well.
So it's kind of comparable to like the face-to-face attack, right?
At the same noise level, I would say this is like bounded at around 20 to 30, whereas for the other, it's like 30 to 40. So it is harder, which might make sense given the separation. Yeah.
Yeah. Thank you. Yeah.
Yeah. No problem. Great.
Okay, thank you very much, Will. And if there are no further questions, I want to thank all three speakers, Mengmi, GA, and Will. And thank you all for your feedback and questions.